I'm Jennifer Gorski, and today I'll be refuting my opponent's claims as well as strengthening the claims that my partner made earlier. So first, my opponent claimed that animal testing is justified because animals do not have the same rights as humans do. And I have a quote from Tom Reagan, who's a professor of philosophy at North Carolina State University, and he said that the philosophy of animal rights demands a commitment to serve those who are weak and vulnerable, those who lack the ability to speak for or defend themselves, and who are in need of protection against human greed and callousness. This philosophy requires this commitment not because it is our self-interest to give it, but because it is right to do so. So that basically means it doesn't matter that animal research benefits us, it's just a cruel thing to do, and it's up to us to prevent it. And since animals can't speak up for themselves, we have to step in and prevent these things from happening. And I also have a quote from Peter Singer, who wrote Some Are More Equal, a book about animal rights. And he said that to people used to dividing up the world into humans and animals, it comes to a shock to realize that the genetic differences between humans and chimpanzees are smaller than those between chimpanzees and gorillas. So, we have a lot of genetic similarities, and that is a reason why we should also protect them, because we are so similar. And it's also been proven that animals have psychological experiences as well. And my opponent claimed that they do not have reasonable thought, and that's why it's okay to experiment on them. But this is a quote from Richard R. Sharp. He has a PhD from the Center of Medical Ethics and Health Policy. And he said that it now appears that most animals have a variety of psychological experiences, including experiences that might be referred to as experiences of pain, pleasure, and other emotional states. So animals are so similar to humans and have psychological experiences, and we all agree that it's wrong to do these types of experiments on humans, so how can we justify doing it on animals when they are so similar to us? I also would like to point out that scientists often use animals for research, even when it's not the best method to use. One review of human-animal correlation in drugs had been withdrawn because of adverse reactions that found animal tests predicted the human side effects in only 6 out of 114 times. So a lot of times, they're not even successfully predicting what's happening in humans, and it's almost like a waste of resources to be testing on all these animals when it's not even providing sound results. And I have a quote from Christopher Andre, who's a member of the Medical Research Modernization Committee, and he said that crucial genetic, molecular, immunologic, and cellular differences between humans and other animals have prevented animal models from serving as effective means by which to seek a cancer cure. Mice are most commonly used, even though the industry's own lab animal magazine admits mice are actually poor models of the majority of human cancers. So they're continuing to test on these mice, even though it's not even relevant. They're not helping the cause anymore. So for the second claim, my opponent said that animal testing is a scientifically sound method, and it is worthwhile because of all the benefits it's produced. But I have a couple of examples that show that it's actually not as effective as they're saying. This is a quote from Kathy Archibald, the Science Director for Europeans for Medical Progress, and she says that even the Handbook of Laboratory Animal Science admits that uncritical reliance on the results of animal tests can be dangerously misleading and has cost the health and lives of tens of thousands of humans. And this is an example of an experiment, and it says, by 1963, prospective and retrospective studies of human patients had already shown a strong correlation between cigarette smoking and lung cancer. In contrast, almost all experimental efforts to produce lung cancer in animals has failed. Because human and animal data failed to agree, researchers and others distrusted the more reliable human data. As a result, health warnings were delayed for years while thousands of people died of lung cancer. And there are some other examples of this, which I will talk about later. But I would also like to bring up the point about how the AWA, my opponent was saying that it is regularly enforced through inspections, and I do agree with this. It is enforced with these inspections, and they do have penalties, but the problem is, like my partner said earlier, 95% of the animals aren't even protected under the AWA because rats and mice are not protected. In 2002, the passage of the Farm Security and Rural Investment Act, also known as the 2002 Farm Bill, amended the AWA's definition of animal to include rats, mice, and birds, with the exception of those bred for research. And I have a quote from the National Institutes of Health that says that most animals used in research are specifically bred for use in medical research. So all these animals aren't being protected, and all the mice and rats that are used in research aren't protected by the AWA. It only protects the remaining 5% of animals that are tested on, and I think that's a huge flaw with it. I would like to briefly go over some alternatives and why they are effective. First, I'd like to define alternative. The word alternative is used to describe any change in an animal test that achieves one or more of the 3 R's. Replaces a procedure that uses animals with a procedure that doesn't use animals. 2, reduces the number of animals used in a procedure. Or 3, refines the procedure to alleviate or minimize potential animal pain. So I have one method that's been used recently as an alternative, and it's basically a chip, and it says this product, this is from an article from MSNBC, and it says the product consists of 2 glass slides. The first, called the MetaChip, has rows of little blocks containing human liver enzymes. The other side, the data chip, contains an identical array of blocks, which depending on the test could be live human bladder, liver, kidney, heart, skin, or lung cell cultures. Sandwiched together, the 2 chips mimic the human body's reaction to compounds. If the cells die or stop growing, it's a sign that the toxin is present. So this kind of gets rid of the uncertainty that there is with animal testing because we're using actual human